Hello everyone, my name is Danna Chacon, and this summer in the Jessica Feldman Lab under the mentorship of Rachel Ng, I investigated the regulation of SPOT5 in C. elegans intestinal epithelial cells. So I guess for starters, what's SPOT5 and why is it important to be studied? Let's start with cell division, specifically mitosis. One regulator of mitosis is the centrosomes, an organelle that acts as a microtubule organizing center, also known as MTOC, by nucleating the microtubules of the mitotic spindle. Centrosomes are composed of microtubule-based centrioles, surrounded by proteins known as the pericentriolar material, also known as PCM, which will nucleate and recruit microtubules. The key PCM component in C. elegans is SPOT5, a spindle-defected protein, which is essential for centrosome function. Organisms depleted of SPOT5 do not survive. What is known about SPOT5 regulation in C. elegans zygote is that 5 key phosphorylation sites regulate SPOT5 function. 2 key sites regulate SPOT5's ability to expand or oligomerize in mitosis. Another 3 sites regulate SPOT5's ability to recruit gamma tubulin and downstream microtubules, shown here in these images, where once these sites are mutated to alanine, the levels of SPOT5 and gamma tubulin are dramatically reduced. While these 5 sites are essential for SPOT5 function in the one-cell zygote, how SPOT5 is regulated beyond the zygote is unknown. My primary question this summer is to ask whether or not these 5 phosphorylation sites also regulate SPOT5 in other developmental contexts, such as the C. elegans intestinal epithelial cells. To study SPOT5 regulation in intestinal cells, we use a tissue-specific degradation system to deplete endogenous SPOT5 only in the intestine and re-express SPOT5 phosphomutants to determine the phosphorregulation of SPOT5. Since SPOT5 is an essential protein, depleting SPOT5 everywhere in the entire organism, it would kill it before even the intestine develops. For worms with SPOT5 degraded in the intestine, I will refer to it as SPOT5 gut minus. We created SPOT5 phosphomutants 5A and 5E, which is a combination of the expansion and gamma tubulin recruitment mutants that are necessary for division in the zygote. While C. elegans intestine at this larval stage should have 20 nuclei, as shown here in magenta, SPOT5 gut minus worms have significantly less, around 7 nuclei on average. When we express SPOT5 wild type, now the worms have 20 nuclei. Surprisingly, when we express either the SPOT5 5A or the 5E, we also saw a perfect rescue of the cell division defects of SPOT5 gut minus. This was a surprising result since based on the results of this zygote where these 5 sites abolish SPOT5 function, we expected that the SPOT5 5A mutant in the intestine to also be non-functional and not rescue SPOT5 gut minus. Seeing that the SPOT5 gut minus have few nuclei, we wanted to see how functional these intestines actually were. To do this, we did an assay called the SMURF assay, where we feed C. elegans with blue color food and use the blue dye to visualize the digestive tract of the worm. As you can see in these images, many cases, the blue food does not reach the entire intestine, showing that SPOT5 gut minus worms have disconnected digestive tracts, and also both inflated and leaky intestinal lumens. Next, we wanted to know if phosphorylation regulates SPOT5 at all. To test this, we mutated all the reported phosphorylation sites on SPOT5 to alanine and generated the 36A mutant. The 36A mutant does not rescue, indicating that phosphorylation does regulate SPOT5 activity in the intestine. Since 36 phosphorylation sites is a lot, we wanted to find a way to narrow down the number of sites from 36 to a smaller number that is more manageable. To do this, I was working on a structure function experiment to determine the minimal components of the SPOT5 necessary for SPOT5 activity, and then mutating the sites within the region. I did this by generating worms with an N-terminal fragment of SPOT5, tapped with a GFP nanobody, which is like a smaller antibody. I will test whether this fragment of SPOT5 is enough to rescue cell division in the SPOT5 gut minus. This is just the beginning of this exciting project. Thank you so much for your attention. Thank you to the Filament Lab and the BSURP program for making this possible. Thank you.